So last but not least, we have Cheryl Nickerson, who is a complete stranger. I've never seen her before, from Arizona State University, who's going to talk a little bit about the past 20 years of her studies doing host-pathogen interactions and understanding how spaceflight impacts both microbial physiology and ultimately what changes the host response for virulence.
All right, so I will try to keep this as succinct as possible because I know you're all excited and waiting for lunch. So my lab has spent the past 2 decades working closely with NASA to understand the impact of the microgravity environment of spaceflight on microbial responses. We focus specifically on microbial responses that are important or could be important for causing infectious disease in astronauts, and also microbial responses that could be important for causing biofouling of onboard life support systems. So what I thought I would do today in that context, instead of doing a single deep dive into any one of those projects, is kind of give you a global overview of some of the major findings and discoveries that my lab has made in that time, and kind of transition then to several of our ongoing studies in that area to help put things in context of how we're trying to help this information advance and enable human spaceflight. So this slide shows you some of the NASA technologies we have used in our studies. We've been very fortunate to have flown multiple experiments on the space shuttle prior to its retirement of the orbiter. When shuttle was retired, we now use Elon Musk's SpaceX technology to fly our experiments to the International Space Station. Now, you might have heard me say I've had my own lab for 20 years. Doesn't seem like it, but I have. And we love doing spaceflight experiments. We've been very fortunate. We're about to fly our 9th one later this year, hopefully. But think about if you had your own lab for 20 years and you could do 9 experiments. You wouldn't have gotten that far, right? So we're very fortunate that we're able to be in our lab just like all of you and your team are 24/7, 365, because we can use this ground-based spaceflight analog bioreactor cell culture technology. It's generically referred to as a rotating wall vessel, and it was designed by NASA scientists and engineers to allow scientists here on the Earth to culture cells in liquid under conditions that simulate the lack of sedimentation and low fluid shear that cells, bacterial cells, human cells, experience when they're cultured in microgravity. But there's one thing I want to add to that. We actually, with our team, quantitated the level of fluid shear that cells experience when they're encountered in those bioreactors that they encounter, and it turns out it's physiologically relevant to fluid shear levels that are in our mucosal tissues. That should matter to you because mucosal tissues like the gut and the lung are among the most commonly infected tissues in the body. Those forces are required to keep those tissues functioning normally. They respond to them. Wouldn't you think that microbes that are in that area, whether they're commensals or pathogens, might actually respond to those forces as well? And so what we find is that regardless of whether we're culturing microbial pathogens or human cells under these spaceflight analog low fluid shear conditions, that we are able to, they exhibit biologically relevant phenotypes that they don't observe when you culture them normally. And that allows us a 2-pronged approach for our research. First of all, we work, and this is what I'm going to talk about today, we work to mitigate the in-flight infectious disease risks for the crew in flight, but we also work to translate our discoveries back down here on Earth for innovative development of vaccines, other antimicrobial therapeutics, and for advances in tissue engineering as well. So again, these are our technologies. I'm going to focus a lot on these in our talk. The ground-based rotating wall vessel bioreactor, and we coined the term low shear model microgravity for these bioreactors. And we use them, just so you know, in 2 orientations. So this allows us to help, they mimic key trends that we see cells, bacterial cells and human cells exhibit in spaceflight. They're not perfect, they're a model, okay? But they have done a pretty good job for us in mimicking a number of the things we see in flight. And so we use them in the laboratory in 2 orientations. One, and Mark described it yesterday, a cylindrical disc that you completely fill full of fluid, your culture medium, you put your cells in there, and you have a solid body rotation. So the rotation of the reactor kind of offsets the cells from sedimenting, and it's going around. okay? And then the cells travel in a low fluid shear environment, okay? And so then we simply reorient that vessel. So now instead of the cells traveling like this through it, as they're not sedimenting and under a physiological low fluid shear, the vessel is very thin through. So if we simply reorient it, oopsie, so that it's now rotating parallel to the bench top, the vessel's about this thick through. So the cells can't stay sedimented. They roll around the bottom and are under levels of increased fluid shear. And of course, we have the wonderful, real, true microgravity environment of spaceflight. So what I'm going to do now, stay with me, is I'm going to go through about 12 to 15 years of our discoveries really fast from spaceflight and work with a spaceflight analog reactor. And we started our work off with the pathogen Salmonella typhimurium. Everybody knows Salmonella. It's among the best characterized pathogens that exist because it's so closely related to E. coli. And it's a major cause of foodborne illness globally. In the United States, it's the leading cause of bacterial foodborne poisoning, and in the United States, it's the leading cause of death from any foodborne pathogen. But NASA cares about this pathogen because it has been a reason that foods that's been destined to go to the International Space Station has been disqualified from getting there. And also, it has previously been found in crew trash before from the shuttle. And we had a lot of experience with that pathogen. So we started off, and our first question was, can spaceflight impact the virulence of this pathogen in a mouse model of infection? And the answer was yes. On 2 independent spaceflight missions, when we cultured this organism in spaceflight, the virulence of this pathogen was significantly increased on the second flight up to 7-fold times. Okay, so what that meant was we got a lower lethal dose 50 that was required for the pathogen to be virulent, and we had a significant decrease in the time to death of this pathogen as compared to identical controls on the ground in the same flight hardware, cultured the same way, except it didn't fly. Okay, and I need to stress to you, the bacteria in this experiment, only the bacteria were flown in space. Why, you say? The mice were infected on the ground. So immediately upon the shuttle landing, we had an identical experiment on the ground. We got the spaceflight experiment. We orally infected mice. You might say, why did you not infect mice in space? You have to ask David Tomko. He won't let us. No, right now you can't infect rodents in space. It's not allowed, all right? So after that, we said, why is this? Oh, and by the way, the bioreactor, I'm stealing my story here. The bioreactor was able to mimic the same increase in virulence we saw in flight. So that was a very important effect for us to have. So we said, why? Why does this environment increase the virulence of this organism? Does it make it more resistant to stresses that your body normally throws at it? Yes, it does. Okay, it makes this pathogen more resistant to acid stress in the stomach, osmotic stress. That's my sound on my computer. I'm sorry, I thought I turned it down. Thermal stress, it makes it better able to resist being killed by immune cells in your body, like macrophages. Now, the one thing the bioreactor could not recapitulate that spaceflight could is only spaceflight provoked this Salmonella strain to cause a biofilm. And to my knowledge, this strain has never been shown to form a biofilm on Earth in the media in which we grew it. You have to really goose it to make it do that, and not in the media we grew it. So then we further said, molecular genetic reason, why? So we profiled the global transcriptomic and proteomic response of this pathogen in response to these conditions. And as you might expect, we saw a lot of genes whose expression changed. But what really blew us away was we have a hotter pathogen. It's more resistant. We figured we'd see a lot of changes in virulence genes that were upregulated, and yet we did not. The virulence genes we saw in the bioreactor, there were no genes that were upregulated, and the bug was hotter. In space, I think there were maybe one that was upregulated and not a lot. But we were able to identify a whole host and suite of genes that heretofore had never been shown to be associated with the host-pathogen interaction, that were being differentially regulated by these low fluid shear conditions. We had a series of clues from previous ground-based studies in the reactor and in some flight work that said we thought we might be able to turn off the increased virulence that Salmonella observed in spaceflight by modulating the ion levels, some key ion levels in the media. We did that, we flew it, we were able to turn off the increased virulence. Back on the ground, use our bioreactor to help us understand which of those ions it might be. And we were able to show that phosphate is an ion that's responsible for allowing this low fluid shear environment to decrease Salmonella's ability to resist being killed by acid stress that's in the stomach. And so I should have said this here. I neglected to do this. One of the things I wanted to mention from our gene expression studies, in addition to boggling our minds because none of the virulence genes were being increased, we were able to identify a master response regulator to about a third of all of those genes called HFQ. HFQ is a highly evolutionarily conserved protein. It goes up through all domains of life. It's been found in about 50% of the bacteria that have been sequenced so far. And this regulated about a third of all of the genes that Salmonella was expressing in flight and in the bioreactor. And what it is, this is a post-transcriptional HFQ modulates the binding of mRNAs and small non-coding RNAs to control post-transcriptional gene expression. Okay, it can turn genes off, it can turn genes on, all right? So we did a classic study, and we said, okay, so let's do the classic structure function knockout. Is HFQ involved in these phenotypes in response to spaceflight analog culture? Let's see, we knocked it out, and we hit Salmonella up with several of these stresses that it normally encounters in vivo, and guess what? An HFQ isogenic mutant derivative of our wild-type strain is responsible for regulating the increased resistance of Salmonella to macrophage stress and acid stress in response to spaceflight analog culture. Okay, now let me take a step further about the HFQ response. We have since shown, as have other investigators, that HFQ in other pathogens are using it to regulate their response to either the spaceflight low fluid shear culture environment or the spaceflight analog culture in the bioreactor. We have since shown this in addition to S. typhimurium and the gram-negative Pseudomonas aeruginosa in flight and in the bioreactor, and in work with Mark Ott and Sarah Castro. This was a part of her, this was a big story of her dissertation defense. I was honored to have served on her committee. She showed that a gram-positive, and this is a MRSA strain of Staph aureus, is also using HFQ to regulate about a third of its genes in response to spaceflight analog culture. And let's not forget commensals. Jamie Foster's work at the University of Florida has found that the commensal Vibrio fischeri, which is synergistically associated with this little squid, when cultured in spaceflight analog conditions, it uses HFQ as well. So it appears that bacteria are hardwired to respond to this low fluid shear response of both spaceflight and spaceflight analog culture. Okay, thank you for that. I should have brought my water with me. I don't have it now, but we're just going to keep going, all right? So based on, oh, thank you, Jack. You are a lifesaver. So based on that, about 12 to 15 years worth of work, what we have done, what I'd like to do now is transition to how we've built upon those studies and have leveraged that work into our ongoing studies. Pardon me. Thank you. And so before I tell you about some of our ongoing studies in the lab to further advance our understanding of the impact of microgravity on the host pathogen interaction and on infectious disease risks, I first have to tell you something. I was kind of naughty. I didn't tell you the other use that we use for the bioreactor in the lab, and that is to bioengineer or develop three-dimensional organotypic models of human tissues that are made from human cells that recapitulate key aspects of the differentiated form and function of those same tissues in your body. So these are in vitro models of human tissues that better mimic in vivo-like responses. And we've done this for 20 years as well. So this is the other half of our lab. And so why do we do this? Okay, tissues and organs in our body function in a three-dimensional context, not as flat two-dimensional layers grown on impermeable surfaces of plastic, which is how they're commonly used to culture. So we have incorporated, we built a wide range of different 3D tissue models, but we focus a lot on the mucosal tissues, the intestine and the lung, and we've incorporated immune cells in them to make them even more like what pathogens and commensals, because we're doing work with microbial commensals on these as well now under spaceflight analog conditions. So with that in mind, you might notice this young lady's picture down here, Jennifer Barilla, who started off as a postdoc in my lab, and now she's faculty at ASU in the same department, and we work very closely and share lab space. So what we've done now is taken those bioreactors and used them in a two-prong approach from some of our ongoing studies. One, to develop these three-dimensional models of human intestinal tissue that contain phagocytic immune cells called macrophages. And then we've taken that same strain of Salmonella typhimurium, grown it, under the two orientations, microgravity analog, low fluid shear, reoriented control, and said, hey, how does spaceflight analog culture impact the ability of Salmonella to colonize more in vivo-like tissues? What does it do, okay? So the first thing we found that it did is it significantly increases the ability of Salmonella to colonize those tissues at every level, at the level of adherence, at the level of invasion, and the level of intracellular survival as compared, look at the blue bars, those are the number of Salmonella recovered when they're grown into microgravity analog conditions, as compared to the gray bars, which are Salmonella grown at that higher fluid shear control. Okay, that result is consistent with Salmonella grown in spaceflight being more virulent in mice. That result is consistent with the RWV also agreeing with the more virulent in mice as well. So the other things we're looking at is we've done multi-omics profiling of both the transcriptomic and proteomic level of both the host and pathogen response under these conditions. Jen is assembling that data right now into a final manuscript, which we hope to have submitted within about a month or so. Following up on those studies, and let me just give you a little bit more insight into how we've incorporated into two new grants, also our 3D tissue models under microgravity and microgravity analog conditions. We recently received two new grants for funding, one of which we talked about immune cells. So Brian Crucian in the back is a key collaborator and co-PI or PI on half of it. I know Mark's the PI and overall PI, and Brian's another PI. There's a lot of PIs on it. It's a lot of fun. So anyway, we appreciate in this first study that infections in our body, as we all know in this room, don't happen by placing a pure pathogen on top of human cells. It doesn't happen that way, right? Infection happens in the context of the microbiome, especially in the gut. So we've been able to obtain astronaut fecal samples from astronauts before, during, and after in spaceflight. And we are now populating our three-dimensional intestinal models with that astronaut fecal microbiome, and then growing Salmonella under spaceflight analog and reoriented control conditions, and seeing how does the astronaut fecal microbiome impact the ability of Salmonella to infect those tissues. We are also looking at studies for modeled viability, colonization, inflammatory responses, and we've got a metabolomic study going and also 16S DNA to look at our microbiome before and after it comes onto the models. And then a separate new study, which is also combining both ground and spaceflight analog culture studies, NASA was interested in understanding the breadth of medically significant organisms whose virulence might be altered in response to spaceflight culture in a manner similar to Salmonella. I just want to tell you up front, in no way do I think, or will it be, that every pathogen is more virulent in space. It's not going to happen. Some will be, some won't be, some won't show any change. So I'm not pushing any panic alarm buttons here or trying to make anybody think these are Andromeda strains. But these are important changes we need to be familiar with and understand for crew health. So what we've done here is we picked 5 medically significant, NASA wanted to know what is the breadth of medically significant pathogens that have either been recovered from the ISS or could get there, okay? And how does their virulence change in response to spaceflight analog culture, including using their impact on astronaut immune cells? So here comes the part with the immune cells from Brian. Okay, and so in this case, we are going to take these 5 different bacterial pathogens, culture them under microgravity analog conditions as compared to reoriented control. We're going to incorporate, oh boy, I'm trigger happy. Yeah, I am. Back one more, stop it. Okay, so we're going to take those 5 different pathogens after cultivation in spaceflight analog and reoriented control conditions, infect our 3D human tissue culture models. Then we are also going to look at hitting each one of those pathogens under cultured in those 2 conditions with different pathogenesis-related stressors that they would encounter in your body. And then we're going to look at their effect on astronaut blood immune cells and profile the wide variety of responses to those cells as well. So we're looking forward to getting that study started. Okay, what I'm going to do quickly now is tell you about 2 of our most recent spaceflight studies, of which we're working on finalizing our work now for manuscripts. The first of which was STL Immune. This flew on Space Shuttle Discovery on STS-131. This was the first experiment to infect human cells in space. Are you surprised to know that we picked human intestinal epithelial cells and Salmonella typhimurium? And here what we did was we simultaneously infected the human intestinal epithelial cells in space. We did microscopic profiling. We did multi-omics profiling of transcriptomics and proteomics. This was only from the host cell response. We're working on finalizing that manuscript now, but I wanted to bring your attention down here to our most recent flight experiment, which was on SpaceX 5 just a few years ago. And I'm sorry, if NASA in the room, somebody mentioned, I think it was Craig, we get so many acronyms from NASA. I have a PIM, a POM, a SOC, and everything else. And so we gave NASA an acronym back, which was Phoenix, which we thought was really clever because it stood for what our experiment was about, pathogen host enteric infection, but it was also where we're from in Arizona. So that made us happy. I don't think NASA thought it was very funny, but we liked it. And so this was the first, remember we can't infect rodents in flight? No, it's a no-no. But this was the first experiment to do a real virulence study in flight. We're both a living host now, not just human cells, a living host and a living pathogen simultaneously, and also profile that virulence in real time. So we did several things here. First of all, we used 4 different media, because some of those media contained what we think might be able to be antimicrobials in that media to see if we could prevent the nematodes from getting sick in the first place. By the way, did I mention that our host is a nematode? A little worm, which is basically a human surrogate intestinal tract from one end to the other. Okay, and so we infected them. We used 4 different media, some of which had countermeasures, and every so often we got little 30-second-minute real-time video clips for the full 14 days. My lab, it took us 2 years. We had 10,000 videos. Okay, so we profiled the infection from start to finish. And what you're looking for in these videos are for the nematodes that are active and happy and swimming around like crazy because they're in microgravity. They're healthy. You're looking for the ones that are straight. And we sorted through and counted through all 10,000 of those in 4 different media types. I'm going to show you a little clip here in a second. We also did transcriptomic profiling from both the host and the pathogen perspective in flight. And what I want to show you is this. And this is a little bit of jocularity that one of my former lab mates has introduced into the presentation. All right, let's do this. And so the nematodes that are wiggling around are happy. They haven't died yet. The ones that are stick straight, those are the ones we count. Okay? Yes, she likes that song. I said, okay, I'll play it for you. But you can imagine now why all my lab members had spots in front of their eyes for 2 years by doing all that counting. So as we've been talking in this conference about humans, NASA pushing humans to live and work longer in space, further away for longer periods of time, obviously disease risks like infectious disease do become something we need to be more concerned about, especially in the context of aspects of the immune system being dysregulated in flight. And that's what led us to publish this study, I guess it's almost 3 years ago now, which was the first transcriptomic assessment of changes occurring in the whole blood of astronauts in response to spaceflight. All right, so this was a set of about 350, 375, I think, stress response genes on microarray analysis. And we had an N of 6, and Rob Plautz is sitting in the audience, and he's a world-renowned expert in low N, and the rest of you, he's going to say, Rob, that's pretty good for astronauts, right? And everybody else is going, no, an N of 6, no. But think about it, the twin study just had an N of 1. When you're dealing with astronauts, it's a different animal. And so we were able to find genes that are encoding proteins involved in DNA repair and oxidative stress, protein folding and degradation, and viral reactivation and immune responses. And you will remember pioneering work from Dwayne Pearson's lab, which has shown that latent viral reactivation happens with astronauts both in long and short duration spaceflight. Okay, so what I'm going to tell you about now quickly are the 2 spaceflight experiments we have coming up. I don't know, Kevin, hopefully SpaceX 19 later this year. That's what they're tentatively slated to fly. The first one is going to now, we're not talking about culturing bacteria for short duration anymore. The vast majority of all microbial experiments that have been done in spaceflight since the beginning of spaceflight have been done for short duration studies. There's a number of reasons for this. Volume limitations, power limitations, hardware limitations. That's the way it is. So in this experiment, we're taking that same strain of Salmonella. Again, we made up our acronym. Again, NASA gave us Micro 13. That just kind of bored us. So we said, no, we're going to call it Evolves, okay? And so this will be, here we're going to be looking at the response of Salmonella to multi-generational growth, several hundred generations, in the chronic long-term stress of microgravity. We want to start to understand the extent and nature of the types of microbial alterations and adaptations we're going to be looking at in spaceflight. So as you might expect, I need to go back. By the way, I should mention, several of our collaborators are in this audience right now, and some of them have just spoken. Curtis Huttenhauer is a collaborator on this experiment, Jennifer Barilla, and Mark Ott as well. And so you might imagine we're looking at a wide array of profiles for this. We are, because now we're looking at heritable changes, not transient changes. So we're doing a genomic profiling. It both changes the A, T, Cs, and Gs, but also epigenetic profiling with methylomics, with transcriptomics, proteomics, changes in virulence, biofilm formation, stress responses, and antibiotic resistance. And to further our mechanistic understanding, in addition to flying a wild-type strain of Salmonella typhimurium, we're flying an isogenic HFQ mutant derivative for obvious reasons. And so now I'm going to transition quickly. I think I have 3 slides left, so I'm going to go fast. As I mentioned, we've also been working with NASA several years now to understand the impact, not just for astronaut health, but on environmental health of the space habitat and vehicle, in particular, the environmental life support system of the potable water. And Mark Ott mentioned this yesterday, but if you look at the ISS, it's not like the Starship Enterprise. It's more like a dorm room, right? And that's the way I view it. And NASA does a really good job of working to keep things clean. Astronauts are continually monitoring things and taking samples. But of course, there's microbes up there. And of course, they've caused problems with mold here on the panel. These are samples of microbes that have been cultured in space and are being held up in space by astronauts showing their pictures. But we all know this. There's no need to go over this slide again. Why should we care about studying microbiology in space habitats? For the same reason we need to understand microbiology of built environments right down here. And we already know in spaceflight, microorganisms have caused infectious disease. They have caused life support system failures with biofilm formation, biocorrosion, system fouling. They have caused biodegradation. They have caused food spoilage. And they've caused release of volatile chemicals. That's what microbes do. And all of these pictures were taken from flight. This in particular is what I'm going to talk about in the next 2 slides. We like to focus on the potable water system on the ISS. And by the way, I know that Mark mentioned this and showed yesterday, this is supposed to be a potable water line with clean water running out of it for which the crew could drink and which they could use for personal hygiene, not a toothpaste-like biofilm. This was on the Russian side of the ISS, so the cosmonauts came over to the US side, because clearly that's not going to work. Okay? And so this is our second spaceflight experiment, which again, tentatively is scheduled to fly later this year. The PI on this experiment is also a speaker at this conference, Bob McLean from Texas State University. And you see some other of our partners in crime, Mark and Jennifer. And so here we're actually going to be looking at the ability of polymicrobial biofilms, polymicrobial bacteria to form biofilms on materials from the ISS potable water system. And we're not just going to be looking at the biofilms formed in the architecture, but importantly for spaceflight operations, the ability of those biofilms to be cleared by disinfectants or not, the ability of those biofilms to cause corrosion, microbially induced corrosion on these spaceflight water systems, because bacteria are great at clogging filters and corroding systems. So that experiment will be flown later this year. And finally, in my last slide, I would be remiss if I didn't cite my postdoc, Ji-Sun Yang. She has done beautiful work. She was fortunate to have been selected as one of the Alfred P. Sloan postdoctoral fellows in a joint call with NASA, and to look at the microbiome of the built environment at the International Space Station. So she's focused on the potable water systems. I like to say her experiment is looking at the social microbiology of the ISS. So she received several years, I think 7 years worth of independent isolates of the potable water when they were sampled from the potable water system. She's received bacterial isolates from each of those years, polymicrobial isolates. She's done an amazing amount of work profiling their population dynamics, their social interactions, antagonistic, mutually beneficial, their social interactions, their metabolism, their nutrient preference. She's done great work, and I'm hopeful when I come back from this conference, I'll have a draft of the manuscript on my desk. But I wanted to give a shout out to her because this is the young generation that's going to be taking the reins over from us. So with that, I'd like to thank all of, most definitely for our sponsors for this work, NASA and the Alfred P. Sloan Foundation, all of our amazing collaborators, both at ASU and around the country. In particular, if there's the NASA Johnson Space Center, NASA Ames. I think I got everyone in the audience that's here for this covered. I do want to mention John Alverdi's group at the University of Chicago, who was instrumental to our C. elegans infection work, and also Villanova University, Jim Wilson, former postdoc, who was instrumental in the first virulence studies we did. And of course, my awesome team, and a huge thank you to the astronauts who so generously donated their time in our work. And with that, I thank you, and I'm happy to take any questions.